Guys, welcome to the video where today we'll be talking about introduction to enzymes. If you guys haven't seen my other videos, I go ahead and do a lot of basic overviews of some of these topics just so it's quick, brief, and very easy to understand. And then I go into more deeper reactions, topics, and stuff that we worry about later. So again, I just kind of want to go over enzymes real quick with you guys so we can just get it over with and you guys can have a quick video to reference for just basic terms. And this is also perfect for anyone that just wants to learn about enzymes and may not necessarily have a boring, crazy professor they have to tend to. So without further ado, let's just get right into it. First, I want to start off basically with what enzymes are. So enzymes are these cool blobs of amino acids, very long amino acids, that speed up the rate of reactions. And they do this by lowering the activation energy. And we can see this in this very first graph that we have here. So to my left, we have an enzyme, and then we have a substrate, right? And as this reaction progresses, we can also see the difference of what happens when there's no catalyst. And no catalyst is just another way of saying no enzyme, because enzymes are catalysts, right? And then with an enzyme. So what we see here is with no catalyst, there's a very high activation energy. And when there's a very high activation energy, it's much tougher for this reaction to go on. It takes way longer. But with the use of a catalyst, it lowers the activation energy. It does not take much energy for this reaction to progress, correct? So that's why enzymes are very important and really cool, because they just make things better. They lower the activation energy, and this reaction can just zip through. It's kind of getting like you're in a race, you're slow, and you have your friend come up and like give you a huge push. This is exactly what these enzymes do. So when an enzyme binds with the substrate, that's called the enzyme-substrate complex. Pretty easy, right? To our right at the end, we do see the result, which is the enzyme unbinding, and then we have the product. So let me just go ahead and blow up what I have. Oh, that doesn't sound right. Blow up. We're not really blowing up stuff. Boom, But I do want to kind of zoom into this right here, talking about more of the structure of an enzyme. So we have something called apoenzymes, and they are just these enzymes that are not active. They're very boring. They have nothing binded to them. So proteins tend to just float around in your body. Some of them are unactivated. How it works is an enzyme isn't going to do anything unless it has to, right? Otherwise, you guys have to remember something. Everything is energetics. A lot of stuff in our body, a lot of proteins, a lot of these reactions, energy is very precious. Why am I going to be active when there's nothing for me to do? So they are left inactive. They're not activated until a coenzyme is present and binds to that enzyme. So enzymes can be lazy too. They're doing all this hard work, speeding up the rates of reactions. However, they also may just be inactive. So again, we have a coenzyme, and what a coenzyme is they're the non-protein parts. So again, an enzyme is a protein, which is this whole thing here. And when it's inactive, it's called an apoenzyme. Then you have a cofactor, and a cofactor helps the enzyme function. So it'll attach to the enzyme, and now that enzyme is up and ready. It's no longer called an apoenzyme. It's called a holoenzyme. And that forms when there is an inactive enzyme that attaches to a coenzyme. So we see here, we have this enzyme plus this coenzyme gives you this, a holoenzyme. So now this enzyme is activated, it's ready to go, and it binds to this substrate, and then it can zip through in the reaction. So what I want to do is kind of go back into a topic that I touched on last video, and this was peptide bond formation. So as we know, we do have a dipeptide that is formed as a result, but I also mentioned how this reaction is reversible, meaning not only can you get this product, but you can go back and form the reactants. And this is done by something called hydrolysis. So remember, in the initial reaction, you have this first amino acid, you have this second amino acid, and then you have this water that's released, which is a condensation reaction, rings a bell, or a dehydration reaction, we like to call it. So I'll just write that there, dehydration reaction, right? And I hope you guys caught this one. What is wrong with this reaction? I'll give you guys 3 seconds. All right, you guys are smart. I don't like these arrows here. Let me explain to you why real quick. One arrow should be bigger than the other. So it should be something more like this, right? And I talked about this last video because the formation of the reactants is more favorable than the production of the dipeptide. And without getting too deep into it, the reason being is that the formation of the reactants is more energetically favorable, because when this dipeptide goes back to these two amino acids, it's broken down by water, right? But that water has the help of an enzyme. Boom. It has the help of an enzyme called pepsin, right? And pepsin is a protease. A protease is a type of protein that breaks down polypeptides, right? And pepsin is an enzyme that does this. So pepsin is a protease. The protease is pepsin. That makes sense, okay? So again, water has the help of pepsin to help this reaction go through. So let me just go ahead and blow up one image here. So this is perfect. So here we have this graph showing the formation of the dipeptide bond. And so we have the products here, and then we have the reactants here, right? So this is A, this is B, this is C, and then the water is D, right? So again, the energy that's required for the formation of the reactants, which is A and B, is lower than the energy needed to form the products, right? So again, the formation of the products is energetically unfavorable. It's high energy, but going from the products to the reactants is a lower energy, especially with the use of pepsin, which is our enzyme, which again lowers the activation energy. Okay, so let me go ahead and just show one more example. And that is with something a lot of us drink, milk, which I am lactose intolerant. We drink milk, goes to our pretty stomach. She has a very nice stomach. Mine looks more like that. But anyway, we drink the milk, it goes into our body, and we have an enzyme called lactase, which breaks down this lactose into simpler sugars called glucose and galactose. If you guys want to see a more in-depth stepwise function and topics of like that Operon, I do have a video out like that. If you guys want to see more in-depth, basically like just how it works, just go ahead and watch that video. But for the sake of this video, I just want to show how the enzyme lactase is responsible for breaking down this sugar, right? So again, we have lactose coming in, takes the two substrates right there, and then it breaks it down into a simpler sugar. All right, so that's the whole basis around enzymes. They're pretty cool. big guys that are very, very important in our body. So again, this is just a quick basic overview. In the future videos, we'll go more in depth, and I hope you guys liked it, and I'll see you guys next video. All right.